RG40 is an organic vegetable extract rich in amino acids and plant kingdom biological factors required for the synthesis of neuronal dopamine receptors. RG40 natural components are organic substances which are easily allowed to penetrate the blood-brain barrier and can access the basal ganglia where they interact with the DNA of dopaminergic neurons. RG40 biological factors are transported inside the dopaminergic neurons, reaching the nucleus where they interact with DNA. DNA in the cell nucleus is the command center where orders to carry out the synthesis of receptor proteins are executed. Shown in this graph is the molecular neurobiology of dopamine receptor synthesis. The process begins in the cell nucleus, where RG40 activates specific genes, which induce DNA to be transcribed into messenger RNA. Messenger RNA then travels to the endoplasmic reticulum, where ribosomes cause the messenger RNA to be translated into partially formed dopamine receptor proteins. The next step is for partially formed dopamine receptor proteins to be transformed into complete dopamine receptor molecules in the Golgi apparatus. Completely formed dopamine receptor molecules are proteins, and these are transported to the cell membrane, where they interact with the neurotransmitter dopamine. As a result of the interaction between the neurotransmitter dopamine and dopamine receptor molecules, the ability of our muscular system to perform movements is regulated. Neuronal dopamine receptor deficiency causes motor disability problems, such as those observed in conditions like MSAP and Parkinson's disease. Most drugs available today to treat Parkinson's disease attempt to increase the concentration of the neurotransmitter dopamine or its agonists in areas of the brain which are affected in Parkinson's disease. However, the activity of these drugs is limited by the number of neuronal dopamine receptors available to interact with the neurotransmitter dopamine or its drug agonists. No matter how much a patient increases the dosage of dopamine precursors like Sinemet or any of the dopamine agonist drugs, if there are not enough neuronal dopamine receptors to interact with, these drugs will not produce any further response at all. This figure is a schematic diagram of a dopamine receptor, showing that it is a protein arranged essentially as a long chain of amino acids. The chain of amino acids forming the dopamine receptor winds in and out of the cell several times. This creates three regions of the receptor. Firstly, the extracellular portions are those parts of the chain entirely outside the neuron. Secondly, the intracellular portions are those segments of the chain entirely inside the neuron. And thirdly, the transmembrane portions, which are the regions of the receptor which reside within the membrane of the neuron. Each and all of the amino acid and biological factors required for the synthesis of this and other dopamine receptor molecules are present in the organic vegetable extract RG40, which is now used by all the PD and MSA patients participating in our studies.